Thank you for my two dear friends and co-hosts. Cyrus, why don't you introduce quickly Nathan and you, and then I can start with my presentation.
My name is Cyrus Khambatta. I have a PhD in nutritional biochemistry. I am the co-founder of a business called Mastering Diabetes, and I've been living with type 1 diabetes now for 22 years. I effectively created a method by which people who are living with all forms of diabetes can reverse insulin resistance, which is the root cause, and look forward to a life with very solid blood glucose control and actually erase prediabetes and type 2 diabetes. I had the pleasure of meeting Isaac about a year ago as part of a mastermind. And Nathan was also part of that mastermind. He was actually the visionary behind it. Nathan is a very good friend of mine himself. He's a certified professional nutrition coach. He's a best-selling author, and he is a award-winning natural health researcher. He's also one of the best athletes I've ever met in my life. He's an incredibly smart human being, and he has helped hundreds of thousands of people understand the true root cause of inflammatory conditions and what they can do about them so that they can live an inflammatory-free life. So Isaac, has become a very good friend of mine over the course of time. And when Isaac first introduced me to the concept of galectin-3, my mind was blown because I said to myself, how come nobody knows this? I've never heard these words before. I've studied biochemistry for 20 years. And when you look in biochemistry textbooks and you go through biochemistry courses, nobody ever mentions galectin-3. Where is this information? And then he opened my mind to a whole collection of information that's purely evidence-based. And he's actually paving the way. He's a true pioneer in this field. So I felt compelled to co-host this webinar with him and Nathan so that we could get this information to more people and really demonstrate just the true power of understanding what this inflammatory pathway is and what you can do about it. Isaac, how you doing today?
I'm doing great. I love keep talking. I'll just relax. It's nice. I won't do as good job as you do. So thank you for allowing me for joining me and giving me the opportunity, and thank you for people who came from your audience, from Nathan's audience, which is the focus of the audience today. And what I thought is that I would start with more of a formal, semi-formal presentation about half of the time, and then we can have a Q&A and dive into different areas where you guys feel that I skipped, because I can get excited and come under the assumption that everybody understands what I'm talking about. And so you feel free to bring me back and allow me to clear. And this is really a path I've been taking for decades. And it's interesting, being a pioneer is not a simple journey, because you are always on your own. When you're a pioneer, it's like you're at the head of the wave. There's only the unknown in front of you. So it's a very fascinating process. And a lot of it is really being a disruptor, is really explaining to people that there are different ways of doing it. And sepsis is a great example. And that's before I start my formal presentation, because a lot of coming from a meditation background, I talk about the survival paradox. The paradox that what makes us survive is what makes us sick. And the survival response really starts with a sympathetic, with the autonomic nervous system response that is uncontrolled, and then it moves into a biochemical response. And this response can be devastating. And the place where it's most devastating, and who would believe me as an outpatient medical doctor, and licensed acupuncturist and research with multiple NIH grants, who would think I would be in an ICU environment discovering that really the peak of this example is sepsis, because sepsis is an infection, used to be thought of as infection of the blood, and it's really not the definition anymore. So with this, I want to start my presentation.
Now, one thing that I can do for you, Isaac, is as you're talking, if you start to get too technical for anybody in the audience, including myself, I might just pause you and ask you to re-explain something. I'm going to try and interrupt you as little as possible because you're the genius, you're the visionary. But just in case there's some questions that pop through, I can play moderator and make sure that people are getting their questions answered. How's that sound?
Thank you. I love question. I love to be interrupted. Easier when somebody else interrupt me and I don't interrupt myself. So it's good. So we're going to talk today about XGAL3, truly a revolutionary approach to resolving sepsis and acute kidney injury. This is really the expression of this multi-decade approach that I came up with now is being more and more accepted. So this is a regular disclosure when we will discuss also investment. You have to give it, and we can really start about sepsis. So it is surprising how many people are not aware of the devastation of sepsis. Sepsis is the #1 global killer, 11 million people, 49 million cases a year globally. Every 2.8 seconds, somebody dies from sepsis. About every minute, somebody dies in the intensive care unit from sepsis in the US alone. And while you have this devastating condition that has more death than all cancers put together, there is an incredible unmet medical need. There is no approved treatment for sepsis, not one. There is no effective treatment for sepsis. Why? Because sepsis is no longer viewed as an infection. Sepsis is viewed as the body's response to the infection, as an inflammatory response, the uncontrolled immune response and inflammatory response, which is the biochemical expression of this survival paradox concept. As such, not only it kills so many people, but it also has an incredible economic burden. Just in the US, $62 billion a year, 40% of intensive care unit costs. And there are about 2 million sepsis cases a year in the US and 4 million cases of acute kidney injury, which is closely related and which makes sepsis worse. And when somebody has sepsis with acute kidney injury, it extends the ICU stay by 50%. And every day in the ICU, not only it costs a fortune, but it means that even if you survive, you may not come out as the same as you came in. And one of the interesting things that people don't realize about sepsis is 20 to 40% mortality in the intensive care unit, but in severe cases, and this can be people that were healthy before, they just didn't catch it on time. It can be up to 60% mortality. And those who come out are never whole again. There's about 1/3 of them will die within a year. 20 to 50% will have readmission into the hospital, etc. So it's a huge, huge global issue, as you can see, with a total global market at $2 trillion. And the addressable market, and I'm concerned that this may not be my final presentation, we'll see, is about 205 billion. So even if you just address 5% of it, it's a $10 billion a year market. So what is this idea about XGAL3? As I will show you shortly, galectin-3 drives the inflammatory process. So the idea behind XGAL3 is to use an acceptable treatment, which is therapeutic apheresis, where blood is pulled out through a machine, the cells and the plasma, the liquid, are separated, and so that's a common process. And here the plasma flows through a special column that we developed called XGAL3, which selectively pulls out just galectin-3. Now you have plasma without galectin-3 coming, uniting with the cells and coming back to the body. And as you keep flowing the blood outside the body, you reduce the level of galectin-3, and with it, you start attenuating sepsis. And XGAL3 column integrates with the, oops, I don't know why it went back. Give me a moment. The XGAL3 integrates with standard apheresis machine for easy use in critical care. And it's a proprietary technology that we developed. It has massive patent protection in 34 countries, 60 patents in multiple families, and it's a platform indication. What do I mean? Galectin-3 drives many of the most common diseases. Sepsis that we mentioned, acute kidney injury, chronic kidney disease, and I'll touch on them later, cancer immunotherapy, and liver and lung fibrosis. So it's not an invention for one single disease. It's an invention that reflects an understanding of what drives some of the most common acute diseases, like the emergency conditions, and chronic diseases. So how does it happen? If we look at galectin-3, galectin-3 is a culprit of, so once the body has a signal of stress, of injury, of infection, galectin-3 gets stimulated, or macrophage, the immune system cells that eat up pathogens, get stimulated and turn inflammatory, and they will excrete galectin-3. So regardless of where it starts, galectin-3 is the initiator of the inflammatory process through increasing IL-4, IL-8, IL-1B, IL-10, IL-6 that many people know, TNF-alpha, NF-kappa beta. This is a classical cytokine storm that we are very familiar with now with COVID. And then it leads to tissue adhesion, tissue injury, and to tissue fibrosis, to dysfunctional tissue, organ failure, and death. And on the other side, it will stimulate the fibroblast and through transgrowth factor beta 1, and fibroblast will stimulate fibrosis. So you end up in those both pathways creating organ dysfunction, either more on a chronic basis or more on an acute basis with inflammation. How does it happen? Galectin-3 is a bus that once it gets excreted, it drives the different ligands to the target tissue. And once it gets there, it creates a pentamer. It creates a lattice formation, a sheet, a cover. And this cover creates a microenvironment where the cells are not functioning in the regular environment. And this can drive cancer, this can drive autoimmunity, this can drive metabolic diseases like diabetes, and this drives sepsis and acute kidney injury. So our goal is to disrupt it. When we disrupt it by removing galectin-3, we are disrupting the pathological cascade. So many people have benefited from my development, invention, and decades of research with modified citrospectin, with Spectasol. It has helped hundreds of thousands of people in multiple conditions. So this is really addressing galectin-3, but in a much faster, much more robust way that a supplement, a drug just cannot do.
Isaac, I got a question for you.
Yes, of course.
It actually comes from Dirk, and that's also a question that I had early on in this process as well. What is the location of galectin-3 production? Is there a specific tissue in which galectin-3 is produced, or is it produced ubiquitously in all tissues?
So galectin-3 can be produced in all cells, but it produced a lot mainly in macrophage, in endothelial cells, and in immune cells. Cancer cells will also excrete galectin-3, and there are galectin-3 receptors throughout our body with a very high density, for example, in the lungs. And that's why ARDS, acute respiratory distress syndrome, that is really what killed the patients in COVID, it's because there's such a high density of galectin-3 in the lungs, you get severe inflammation. So it's really the immune system that upregulates it by getting a signal that something is wrong. So we have galectin-3 release. It's released into the bloodstream during sepsis and AKI, and we have extensive data on this I'll show you in a moment. And then it drives the inflammatory process and the fibrosis on a long-term basis, causing later on organ damage and death. But it's also a prognostic biomarker, which I will show you some remarkable studies in a moment, meaning the level of galectin-3 when you come to the ICU with sepsis will determine the outcome better than any other marker. So because it addresses inflammation and fibrosis, beyond sepsis and sepsis-associated acute kidney injury, XGAL3 can treat many galectin-3 driven conditions. And our roadmap is once we get approval and we establish the use of the XGAL3 in sepsis and sepsis-associated acute kidney injury, then we are going to focus on chronic kidney disease, 37 million Americans, okay? Almost $100 billion annual expenses. On adjuvant to cancer treatment, especially immunotherapy. Immunotherapy does not work if galectin-3 is elevated. And we are now about to engage in a very large diagnostic trial to confirm this and help us select the patients. Right? We hope to test up to 300 patients who are coming into immunotherapy in cancer and check their galectin-3 before every treatment from the beginning and see how it relates to the response. We know that galectin-3 increases expression of PD-L1 and lowers the efficacy of PD-L1 inhibitors. And galectin-3 drives fibrosis in organs like the liver fibrosis, NASH, and lungs, pulmonary hypertension, idiopathic pulmonary fibrosis. These are conditions where galectin-3 etiology is well known. These are huge markets with, unfortunately, 100 million Americans involved in this. So really, we are taking a known therapy that is used in very rare conditions, unfortunately, and we are making it a therapy for the most common conditions where there are no treatments. That's the value of this. That's why I'm not spending my time meditating like I planned, but I'm really committed to really making this happen, because it is so pivotal to find a solution for this. So where we are from as a company and our competitive advantage is that the galectin-3 apheresis has rapid effect. We see in large animal studies, in pig studies, which are similar to humans, the effect within hours in inflammation in general, and we are seeing a major attenuation, a major decrease in severe sepsis process within hours. Within hours, to be conservative. Literally, within an hour, we already see things shifting. And the importance of this is that rarely does a sepsis patient die right away. They all get resuscitated. They die later on from the organ damage because of the severe inflammation. So the quicker you cut this inflammation, the quicker you're going to protect the organs. It is economical because it costs less than biological, and the development is much cheaper. We have a massive patent protection in multiple families, including patents that are being filed all the time. It has a long-lasting effect from the point of view that we can repeat it as many times as we want. And the more you repeat it, the more the body is under low level of galectin-3, the better the results will be, which is not the case many times with drug. It is safe because you are treating the person outside the body and you are removing a very select protein. You're not depleting the body. You don't have metabolite like with drug. And it's profitable from the point of view that it's much cheaper to develop compared to drug, a fraction, and getting to the market is much faster than a drug. So the key milestones of Eliaz Therapeutic that I'm very proud of is from the point of partnership, we have a solid collaboration for a decade with Terumo BCT, a renowned global leader in apheresis with presence in close to 200 countries. And we are collaborating with another very large multinational diagnostic company. We have preclinical and clinical evidence. We have successfully demonstrated, and I'll disclose some of it. We are about to submit the manuscript. We successfully demonstrated proof of concept in both small and large animal studies in sepsis. We are now preparing for our GLP safety study, for which we got an NIH grant, and then we hope to file for clinical trials in mid-2025. And we are backed by 6 compelling scientific publications, including one upcoming manuscript that is going to be really a landmark manuscript. We have a solid IP, which is often unusual for a startup, where we first of all focused on creating intellectual property. So what we do is protected and it's not easy to steal. And we are at the final stages of prototype development before starting clinical trials. So now I want to step for a moment and talk about talk a little bit about galectin-3 in critical care in the ICU. So this is a landmark paper that was published in 2022, and if it was published when we submitted our initial breakthrough designation in 2021, it would have been approved. This is a study, a high-quality study published in critical care, 2,076 patients, multiple centers who came to the ICU. Not all of them have sepsis. That's the beauty of it. The galectin-3 addresses many of these other diseases where there is crisis. About 40% are sepsis. And what you see, that at the day of admission, the level of galectin-3 at the day of admission, and look at the high significance statistically, okay? Okay, smaller than 0.001. Okay, that's the level of statistical significance, and barely any overlap. The level of galectin-3 at day of admission will determine which patient later on will get a major adverse kidney event, which patient will get acute kidney injury, and which patient is going to live, and which patient is going to die. Remarkable importance. And when we analyze it deeper, and we develop the galectin-3 into 3 groups, we see that in the lowest group, 5.2 to 15.9, which is almost the average in the population, you have about 10% mortality. Between 15 and 27, it doubles. And between 27 and higher, there are very few at 114. Most, the average is like 33, 34. You're almost at 40%. When you look at the combination of acute kidney injury during the stay, ICU stay, in galectin-3 levels, low galectin-3 with no acute kidney injury, under 10%. No acute kidney injury, okay, there was no kidney damage, but galectin-3 is above median, 31%. The patient got acute kidney injury, okay, there is kidney damage, but galectin-3 at admission is low, better survival than galectin-3 high and no acute kidney injury. That's the significance of galectin-3. And both of them are high, you're reaching 33%. And this is very critical. This is the levels of severity of acute kidney injury. No acute kidney injury, and this is based on damage to the kidney and the increase in creatinine, etc. So no acute kidney injury, that's average 16.6. KDIGO level 1 is 23.6, KDIGO level 2, 31.3, and KDIGO level 3 is 38. But these are the patients who will end up needing dialysis, and many of them will not make it.
Isaac, I got a couple questions for you here.
Of course.
If you don't mind me cutting you off here.
Yeah.
Could you go back to the previous slide?
Yes.
Okay. So in these 3 graphs here,
Yeah.
the 3 panels, the very first one says no make and make. So that's basically acute kidney event. That's what AKE
Right,
says. Right. Okay. And then we have no AKI versus AKI. So AKI is acute kidney injury, correct?
Right, it's different. An event is a certain increase in creatinine. They're different. I don't want to get into this into category.
Mm-hmm.
And acute kidney injuries where there is a certain level, kidney don't go to normal. There's a certain definition, you know. Yes.
Okay, fair enough. And then on the right-hand side, we have basically survivor versus non-survivor.
Right.
So here's my question. 2 questions. Number 1, what is the time period from which you can go from an acute kidney event or acute kidney injury to death? Are we talking days, hours, weeks, months?
This study is a 28-day study.
28-day study. Okay. And this was again in pigs, correct?
No, this is in human intensive
humans.
care unit. Yeah, this is an intensive care. This is a human clinical trial.
Thank you. Okay.
4,026 patients.
I think I missed that.
No, this is human. This is people coming to the ICU.
Okay.
That's the significance. This makes galectin-3 shine as a predictor and a determinant of what's happening. And you will see the result in a moment. So some of the work that we had done in critical care before the apheresis, where we still didn't have a column for rats, so we used MCP. We did 1 study on sepsis patients and 1 study on patients who have perfusion injury. They have no supply of blood for a short time. That's what happens when you have cardiac surgery and you are in a heart-lung machine. So basically, if you look at this, and this is something that happens during sepsis as well. So you look at these 52 patients, their level of galectin-3 when they come into the ICU after surgery, okay, so about 1/3 of them had AKI, which is very common, and it resolves many times. And this is why people can get bypass, and they found that the arteries are blocked, and suddenly they never come out of the ICU. So you can see a very significant difference in the level of galectin-3, okay, and who will get AKI later on, not at the time of admission, okay? You can see there is barely any overlap, okay? This is 25 to 75%. So the 75 percentile is much less than the 25th percentile in people who got AKI. And same thing you see in sepsis. In sepsis, about almost half of the people had AKI, and you can see a dramatic difference in the level of galectin-3. Also, galectin-3 at the time of admission will determine who is going to die from sepsis. And with this study, we also did a study in rats where we showed that when we give them galectin-3 blockers for a week before, then we attenuate. There's a dramatic decrease in kidney damage, in interleukin-6 rise, and in death. I just don't want to take all the time. It's not a scientific presentation. I'm just covering it briefly. And these were published in very good critical care journals. So a little bit about our journey with apheresis. So our initial study was done with Mass General Hospital, Harvard, with Dr. David Sachs, one of the fathers of immunology who discovered HLA2, the self-recognition system. And then we'll show you. So this was done on a skin inflammation model, which is David Sachs' specialty is on xenograft transplant and transplanting organs from pigs into humans. And that's a model to look at rejection. So we put a catheter in the animals, then we give this injection of an irritant, which is a modified infection. Okay, TB, it's really tuberculosis bacteria, bacillus modified, and it creates inflammation. We do it on day 0, and then we give apheresis on day 1, 4, 9, 15, 18, 22, 25, with a column to remove galectin-3 and without a column. This was our initial study. And you can see the dramatic differences at day 10. Terrible ulcer, unfortunately, in the control. No ulcer in the treatment. Terrible inflammation of lymphocytes and tissue, which is pretty much normal. And what we saw here is we saw an immediate response. We saw that immediately the circulation improved when we started the study. And that's the first time when I had this click, wow, this is going to be good for sepsis. So some of our work, that's the work we just presented in the ICU, that is very important. 87 ICU patients with septic shock, severe sepsis, 27 normal controls, and we can show that at the time of admission to the ICU, the level of galectin-3 is much higher in patients with sepsis than in normal population. Serum creatinine is not different yet. And interleukin-6, of course, is different because it gets reacted very fast. And now we come to our specific studies. That's the first apheresis study was done in sepsis. We developed a special antibody just for red galectin-3 for the whole project. And with this antibody that we've proven effective in removing galectin-3, we used whole blood apheresis because the red circulation is too small to separate the plasma from the cells. And we treated 10 rats with apheresis with the antibody, with galectin-3 depletion. And 9 rats had a sham apheresis. Only 11% survived, 8 out of 9 died, only 1 out of 10 died, 9 out of 10 survived in the apheresis. Very dramatic results. And this moved us to the main model, which is a lipopolysaccharide-induced sepsis. Lipopolysaccharide is a compound excreted by bacteria, but also excreted through the gut in time of inflammation, and it drives sepsis. So this is a very aggressive sepsis model where we implant the catheters for the procedure. The animals are in an ICU environment. They are sedated, they are intubated, there are 5 ICU doctors working on each of these animals. It's a remarkable setup. You'll never get it. This is not like a technician done. It's actual board certified intensivist. And then we give lipopolysaccharide. The animal's blood pressure drops, we stabilize them, and then we start the acceleration phase. 10 minutes at 6 microgram kilogram an hour, 12, 24, at 45 minutes, full-blown sepsis. Full-blown sepsis. Animals that don't get treated except support, 70 to 80% will die in 24 hours. This is a very large study on 31 animals. We start apheresis 10 minutes into this. Okay, we already did the initial one. It's what will happen in the hospital, in the wards. We rush them to the ICU. They're getting worse. We get them set up. We start the apheresis. And here is what happens, okay? And again, I can't show all the details because we are about to publish it. In the control group, only 4 out of 15 survive. In the active, which is between 20 and 30%. In the intervention group, 11 out of 16, about 70% survived. And those who didn't survive really didn't survive because the column got saturated. And now we have an improved column, so we know we'll have a better survival. So very dramatic. But as important, we saw a significant difference in the parameters of severe sepsis. Extra lung water index, that really, it's what causes ARDS, acute respiratory distress syndrome, where people die from in sepsis. Lactate, which causes acidosis and cellular dysfunction. And we also, these animals that got the treatment didn't require very little IV fluid and did not really require vasopressor support. Their system, their blood pressure did not collapse. It's a very detailed study, and you will see when it's published, it's really going to have a big impact. But this is just to show you the impact on the organs. After 24 hours, when the experiment ends and the animals are sacrificed, we take from a select animals that survived, we take samples from the organs. So you can see the baseline of the heart, normal tissue, and you can see what happened in the control, in the hemapheresis, where it's done without a column, just circulation. Total structural damage to the heart compared to a normal tissue. Same with the liver, very similar with the lungs. Look at the lungs completely filled with lymphocytes and fluids, and look at the kidneys, right? There's a minor change compared to major swelling of all the of the nephrons. This is what will cause over time the organ damage and the death. And that's what we are preventing, and in such a short period of time. So a little bit about going now back to what we plan. We hope to get approval by 2028. And this is our projected sales. These are projections, of course. And these are the initial market, the additional market we are planning to go: chronic kidney disease, adjuvant to cancer treatment, and other fibrotic conditions. We have a very solid team. Anat Stern has been the CEO of Econogenic, which I sold successfully, very successfully last year. She's been working with me for a decade. Glenn Cherto, a clinical director, was the head of UCSF nephrology, head of Stanford Nephrology, also an investor, and he has over 1,000 peer-reviewed papers, very few doctors. He's a world-renowned nephrologist and one of the leaders, and we have a very strong scientific team from the apheresis world, Patrick Moriarty and Jeffrey Winter from the Mayo Clinic. We have Ron Wald, which was the principal investigator of a very large acute kidney injury and sepsis study and the use of dialysis, over 3,500 patients. So he's going to help to run the multi-center clinical trial. And Daniel Keitzman, who is urological oncologist, published the big paper on MCP and will collaborate on the diagnostic and therapeutic in cancer, which we are planning to start in parallel. We are geared towards training intensivists in the intensive care unit and nephrologists on this treatment. And our initial focus is the USA and Europe, and then we're going to move to secondary markets. And really, we have a very robust IP protection. It's kind of my expensive old hobby. We have 4 major patent families, plus other ones, one for galectin-3 removal in general. So as long as we remove 10% galectin-3, we can do anything else. That's all. So it's a very broad patent, device patent on using different patents, removal of other compounds, combined treatment for other conditions, and multiple other patents on including removal of COVID, removal of viruses. We filed for sepsis and other patents. We always have patents on file. In fact, we're about to file another patent, which I can't disclose in about a week. I'm reviewing the final application. From an exit strategy, we have good relationship with the companies who may be the companies who will buy us one day or collaborate with us. If it's Terumo, Fresenius, DeVita, Abbott. For some reason, I think this slide is not, this is not the final slide that I prepared. We have a relationship with Abbott and B. Braun. All these companies, we know them, we're in touch with them. And we also have a lot of room for working with pharma once we show that it's also good for immunotherapy. Really, this webinar is really to invite people to join us and invest through our crowdfunding, through this concept of community funding, where you can invest a very small amount or a large amount, of course. And the reason that we feel it's a special opportunity, because it's a promising technology, it has an encouraging, solid proof of concept in a large study, 31 pigs, and in rats. It has a high safety, easy adoption, because apheresis is common in hospitals. It's a platform technology, and the time to market is quicker than with drugs. What we're offering now through WeFunder, which is one of the main platforms for community funding that has raised over $600 million for over 500 companies, is a valuation cap of 49.5 in general and discount 10%, and maybe I'll explain it in a moment, an early bird cap of 30.96. So what does this mean? It means that when you invest in a form called SAFE, which I'll explain in a moment. And in this form, when we issue shares in the future, the highest price that you will pay before the additional money that is being invested is 49.5 million, regardless of what the price will be. And you'll get a 10% discount from the price. But for friends, people from our communities and what we call early bird, we have given a special 20% discount on the cap. So currently, you are coming in at a cap of only 39.6. If the company raises at 44 million, then it will stay 39.6 because it's 10%. If it raises at 60 million or at 100 million, you stay at 39.6. And the minimum investment is really symbolic. It's more for people to join. But of course, without money, we can't make this happen. And this is, of course, it's a startup. There are always risks, but there are also potential upsides. And this is a link to wefunder.com, Eliaz.therapeutics, where you can invest. So a little bit for people who are not familiar with WeFunder, it's a well-known and reputable crowdfunding platform for startups and small businesses. Many medical device companies use it. It is registered with the SEC, member of FINRA, and has strict regulatory oversight. And it's open to both accredited and non-accredited investors, so anyone can invest. And the form of investment is called SAFE. SAFE is the most common investment vehicle, simple agreement for future equity. All companies pretty much use the same standard agreement. That's why investors like it. It's straightforward, and it's more acceptable than convertible notes that used to be used. And basically, the principle is you invest now, equity later, but your conversion terms are predetermined. So you know what is the highest price that you can pay. So you go in at the same price that an equity round where for larger money the investors get, but there's an additional discount. This is a little bit about the presentation. And now that Nathan and Cyrus are joining me, please feel free to ask me questions and anything that you want to.
So, it's funny, as you're going through this data, I've seen some of this data before. And I remember when we first started communicating about it, I was shocked because in the, 20 years that I've been in the world of biochemistry, it's rare that you come across data where there's such a large separation between the treatment and the control group. Usually there's like a tiny little, maybe like a one, like a 0.05 separation, which is like, a small statistical significance, and then every once in a while you get like a slightly larger. But your data, both in humans as well as pigs, shows that there's a very large separation between those that have been treated and those that have not been treated. And when you get such a large separation between the groups, and you see this repeatedly over and over and over and over again, that really shows you that within a very short period of time, you can induce very large physiological changes, which result in death or no death. Like the final endpoint is whether or not these animals and or humans have the ability to stay alive, right? And sepsis is an acute condition that has to be dealt with in an acute manner. You cannot waste time. And when you see such an effective treatment that can elicit such a large biological change within 30 minutes or an hour or 2 hours, you kind of have to stop and take a look at it and say, wow, what is actually happening behind the surface here? So that's a long-winded way of me saying these changes are big. These changes are very big.
Yeah, definitely. Nathan, do you want to add anything?
Yeah, absolutely. Thank you, Dr. Isaac, for doing this presentation for our community. I know there's a lot of people here who know you, and some people probably this is their first time meeting you. And for those who don't know, Dr. Isaac Eliaz has been in the research field as a pioneer, uncovering this chronic inflammatory state, which is basically created by galectin-3 for over 3 decades. He's been doing this for around 30 years now, and is a very accomplished, published scientist, one of the most respected medical doctors in the field. And the reason that I got involved and I decided to invest in this is threefold. Number one, I trust Cyrus dearly, who's a good friend of mine, and we were at I believe we were at a retreat, and he told me, he said, "Hey, Dr. Isaac has this thing he's been working on for years, which could save millions of people's lives. I'm thinking about investing in it." And I said, "Well, tell me about it." So we went and met with Dr. Isaac, and he showed me some of what we just saw here. And when I saw the potential of literally the potential to save millions of people's lives with what I would consider probably the most, non-toxic, I would even, call it almost a natural approach in a hospital setting that we will probably ever see in our lifetimes, right? That it wasn't another pharmaceutical that had all kinds of potential damaging side effects, but in fact, it was something that could already hook up to the technology that's already in hospitals and literally potentially save millions of people's lives. And then the ability for all of us here to be an early contributor, an early investor in this. It was, at that point, it was just a no-brainer. Knowing Dr. Isaac, his background, getting to know him the last few years, the research and published science that he's done, and literally has been a pioneer in this field. And then looking at what's already been done to this point, a lot of the legwork's already been done, right? These published studies are already showing how effective this can be. And I just thought, you know what, this is an opportunity of a lifetime. And I have to tell you this, I don't think I told you this, Isaac, I literally, probably a few months before that, I was kind of in my prayers and meditations, praying to God, saying, look, I don't know, a whole lot about investing. I played around with some stocks and some bonds and some this and some that and some real estate and some, things here and there. But I would love to be able to invest in something that had a massive upside that actually really helped people with people that I trust. I was literally asking for this. And then Cyrus is like, "Hey, I got to show you something." And then I sit down with Dr. Isaac, and I was just like, this is it. Thank you, God. So for me, it's a no-brainer. And so, I wanted to invite my community here to be able to hear about this and have the same opportunity that I had to be able to be a part of this, to be a part of the solution in a non-toxic, very effective way, and be early, which means those of us who are early in this are obviously, the ones who will benefit the most, right? You wait later down the line, you're like, yeah, that's, maybe I'll wait a year, see what happens, 2 years, see what happens. If and when this goes to market, which it looks very likely, right? And then we'll probably be bought by one of these huge companies and then, implemented into hospitals everywhere. which I love too, all the other potentials from it. You know, the potential for cancer, the potential for a lot of the other issues, you know, chronic inflammatory issues that it may be able to support with, and seeing that massive potential, being in it at this stage, you know, everybody here, I just wanted to be able to have the same opportunity that I had. And so, Isaac, I want to say thank you for literally dedicating your life to this. And I'm excited to see it come to fruition. So thank you. Thank you for inviting us to be a part of it.
Yeah, thank you. You know, yeah, it's really striking. That's for me, you know, it really changed my life. My plan was after echogenics to just focus on meditation, on transforming the survival through meditation. But this is, as my daughter told me, who is a doctor, and spent time in the ICU, said, Daddy, this is too big. It's going to change medicine. And she's right. So really, maybe let me clarify some questions that people have. So this investment will convert into shares, and it will convert into preferred shares. Many times companies give common non-voting shares. You're getting the same shares that the big investor who comes in will get, but you are getting an additional discount or minimum of 10%, and you're getting a cap, which means a very large investor can put $20 million, and they may pay your price, or maybe they pay 3 times, may pay 100 at a valuation of 100 million. You are capped at 39.6. So yes, for the person who asked, it's going to convert into shares, or if we get very lucky, we get a lot of grants, and this community raise goes very well because it's in multiple stages. And the only time you get your shares is when we sell the company. Then there is no dilution, and then you know, and then of course. So it is a startup, and that's why there's always a risk. For the people who ask me if it's like investing in oil, it's different. The main difference is that when you invest in oil, you are supporting something that's going to get people sick and make them have cancer and make them more susceptible for sepsis. And here you're investing in something that's going to save people's life, including people who got damage from petroleum products, as a side comment. But generally with a startup, there is a greater risk and there's a much greater upside. That's the regular, and of course we can't make any promises. We're not allowed to legally to really do it, but we have de-risked a lot of the things. We have a massive patent. We have, I mean, you can learn from the fact that massive collaborators from this industry are with us, despite the fact that they usually will not collaborate with a company before they get FDA approval. And the question, will the established traditional medical system try to block? The other way around. I just presented in a prestigious international sepsis conference with really the leading sepsis doctors. And the consensus is that filters are the wave of the future for sepsis. There is a consensus by one of the pioneers. Okay, so this kind of approaches are going to be what will make the difference. And for people who ask about this compared to other apheresis, you can combine galectin-3 apheresis column with other methods of apheresis. They cannot combine it because they will be violating my patents, but my patents allow me to use this with any other columns. But this is very different from general LDL apheresis, or from, of course, from plasma exchange and from anuspheresis. These other systems are non-selective systems. Here, we are taking out the needle in the haystack. And it was, as David Sachs, before we did the studies, told me, Isaac, you're asking for a lot. And then after he saw the results in the pigs, he sent me an email, you really got something. We found the needle in the haystack. Okay, the pigs in 45 minutes, they simply don't go into sepsis. They simply don't go into sepsis. There is no need to resuscitate them. There is no need to give them fluids, except to balance them because they are on a respirator. There is no need to give them pressors. There is no increase in extra lung water index. You'll see it when we publish the paper. It's mind-blowing. So we actually found something that shuts down the survival paradox at its worst. And that's very exciting. So I really want to thank all of you for joining me and my dear friends, really, and thank you for your support and your trust, and Nathan and Cyrus. And I want to invite people to go to WeFinder and join us. I do believe it's a good investment. I can't endorse it because legally there's a lot of restrictions. But I've put a lot of my money and time and energy into it, and I wouldn't do it. People who know me know. I mean, I wouldn't spend the time if I didn't truly believe in it, and if I didn't see the potential in my history of being successful in what I do. And somebody asked me, don't you have a good indication of this needle in the haystack from your work on Pectosol? Of course I do. This is how I got the idea. I realized, my God, I was sitting getting a lecture about certain plasmapheresis in Germany in 2011 with Ralph Moss. And I wrote him a note and told him, Ralph, I just made the invention of my life. And I gave him the note. Of course, but this is so dramatic. We're able to deplete immediately. We are in our new column. We are seeing that there is no galectin-3 under the column, even at fast enough of a flow. So in the animals, we don't need to deplete a lot. It's enough that we deplete 30-40% compared to animals who are not being depleted, and there's a dramatic change. If you look at galectin-3, it doubles, it triples. Why? Because it starts the process. You look at interleukin-6, it goes up 1,000-fold, 10,000-fold. It's a downstream molecule. That's why removing it does nothing. It's too late. It's like stopping a waterfall with a bucket. We're literally shutting the process. And so that's really, but yeah, I mean, Pectosol is, with its close to 100 published papers, is a testament to the power of galectin-3. So yeah, it's a good question. So thank you again, everybody, and thank you, Nathan and Cyrus, and thank you to your communities. I didn't maybe present myself enough for people who don't know me, so I apologize. And the minimum investment is $250, definitely. And there is, I think that on the chat, we sent you the link, and we will send it to you again in the follow-up email. And please join me, because I believe it's a good investment, but mainly because together, I need help of other people. Together, we can change the outcome. And really, if you think about it, we are starting from a concept that is so holistic. Understanding that the root of disease is our survival drive. Okay, this is a very philosophical principle that really drives vast levels of our reality. And we're taking it to the more scientific level, to a patient in the ICU on a respirator who is about to die, and turn it around. And like I tell my friends, what I want is to see the first patient turning around. Then there are really good business people that will take it to where it is. That's what I want. I want to see the first patient, because I didn't tell you, my father died in an hour from such a process. It was the biggest success of immunotherapy. In 1 hour, he was in Cornell, and he had hypernephroma with amazing response and too strong of an immune system. He got a strong immune response, the lung filled with water. He was gone in 30 minutes.
We're all here because we believe this is a great investment for humanity, for the health of human beings, and for all of us financially. That's why we're here doing this for all of you, to let you have the same opportunity. But if you think this is a great opportunity and you choose to invest, or you think it's a great opportunity, this is something we want to share. On that WeFunder page, you can share this with friends, family, social media, anybody who you want to share it with, because the more people we get involved, the more funding that comes in, the more likely that this is going to happen even sooner, right? So this is not something that you invest for yourself and think, okay, this is just for me and my family. This is something we want to share with the world as quickly as possible, and that's something we all can do to contribute towards this. And as you said, a totally holistic solution. It's just like the person who said, well, will the medical establishment try to stop this? Of course not. They want to buy this so that they'll make the money from it, because they'll be able to use it and make billions and billions and billions of dollars from it, which is fine, right? This is something that would be great if a large, and that's probably most likely what will happen, from what I understand, right? Where a large, maybe a pharmaceutical company or a large medical company will come in and purchase this and then be able to be the long-term profiteers of it, which is wonderful because it's less drugs going into people and causing, side effects and potentially massively healthier solution to save people's lives.
Andrew asked a question. He said, Cyrus, how can Alma Green play a role in this? The way that you can play a role in this, that I felt compelled to share this with the Alma Green audience. We have thousands of people in our audience, and I wanted to make sure that we could get this information to as many people as were interested. So my goal is to just try and spread this far and wide. So Andrew, if you're interested in the opportunity, fantastic. If you're not interested, but other people that are, fantastic. Share the presentation with them, share the WeFunder page with them. No problem. This is life-saving technology. And, we want to make sure that you guys have an opportunity to participate early before this becomes, a very large technology, and it's too late.
Thank you. Thank you very much. Really, I really appreciate it. And I really thank you for taking the time to listen to my journey and my focus in life right now.